Alzheimer's disease genetic testing today really is largely confined to research and pharmaceutical company activities, where people are testing essentially for one gene, the APOE gene, so-called apolipoprotein E gene, which comes in several forms. And as you may know, if you carry the E4 form or allele, then you're considered to be at relatively higher risk of developing Alzheimer's disease versus someone who doesn't. So that's the kind of state of the art. Some companies look to stratify for people who carry E4 and then enrich their trial populations with those subjects. But really, clinically, it's not used routinely at all, with one or two, minor exceptions. And that's because it really doesn't tell you very much. I mean, lots of people that carry the high-risk APOE4 variant or allele actually don't go on to develop Alzheimer's disease or suffer any symptoms. Conversely, about 40% of the Alzheimer population is made up of people who don't carry E4 at all, so the so-called APOE3 homozygotes. And so clinically, it's really not proven very informative and is therefore not used routinely. So APOE is the sort of state of the art today, but really there's a huge deficit in performance that needs to be addressed before genetics are more widely adopted, I think, particularly in clinical testing. The genoSCORE has really been developed to close the gap between APOE and the full genetic components associated with increased risk. So actually what we do is we take a saliva sample from a kit sent to either the physician or even the patient in their own home. The saliva sample is sent back by the post, DNA extracted, and then we run a genotyping analysis, which looks at 112,000 different genetic variants across the genome, all of which contribute a little bit of risk or protection against the future development of Alzheimer's disease. And that's a far more informative test than just looking for APOE alone. APOE is one of the components, one of the 112,000 components we look at, but it's just one. And we add together the whole of those genetic variants, the whole picture of those variants, in a weighted algorithm to generate what we call a polygenic risk score, which is a comprehensive assessment of genetic risk. And that test takes about 2 to 4 weeks from when the sample is received in the lab to when the report is generated. And that report is sent back to the clinician who's looking after the patient. And the clinician then talks the patient through that report and things that he or she can do to reduce future risk, because we can test for risk in individuals who may have early symptoms, or indeed who may still be pre-symptomatic. They may be coming forward for testing because they have a family history, a mother or father, a brother or sister who's had Alzheimer's disease, and they want to know whether they may have inherited any form of risk as well. So we're talking here really about a test that can be used to look for future risk in people before they develop symptoms, or who have early symptoms which may be unclear as to whether they're Alzheimer related or not.